Welcome to this special Choices and Challenges teleconference, the Genie in the Genome, the Human Genome Project. From Virginia Tech in Blacksburg, Virginia, here is the director of the Choices and Challenges series, Dr. Doris Zalin.
Thank you for joining us today. For 7 years now, Choices and Challenges forums have examined the major advances being made in science and technology. And at all of the forums, we paid particular attention to the ethical and societal consequences that these advances have created. Today, we're focusing on what may be one of the most dramatic scientific ventures of them all, the new effort called the Human Genome Project that is seeking to find the location of every human gene residing on the human chromosomes and to determine the precise chemical structure that characterizes each human gene. Our genes are composed of a substance called DNA. A gene is really just a length of DNA which contains the code, the instructions necessary to help bring about each of our physical and chemical features. The code can be found in the order of the bases within the DNA molecule. There are 4 types of bases in DNA: adenine, guanine, cytosine, and thymine. They are generally known by their first letters, A, G, C, and T. The specific sequence of these bases provides the blueprint that determines for each of us our own physical and chemical characteristics. The Human Genome Project is expected to yield extremely valuable information about our genes, enabling us to learn exactly how genes function, how they help us become the human beings we are, and how, on occasion, genes can malfunction, causing us serious health problems like cancer or heart disease. At the same time, the Human Genome Project and the information it produces may also raise some extremely difficult questions. Our Choices and Challenges program today is designed to examine the Human Genome Project in some detail, and what is most important, to identify some of the key ethical, legal, economic, and social concerns that it may raise. We have the unique opportunity, if we can identify some of these concerns early enough, to take action now that may avoid problems later on. We've invited an outstanding panel who will be sharing their insights and views with us today, but we need to have you share in the discussions too. For those of you in our distant audiences, there is a toll-free number that you can call. It's appearing on your screen now, and we will repeat it throughout the teleconference so that you can have the opportunity to participate. We look forward to hearing from you.
Hello. To those of you who expected to see Robert Bazell of NBC News moderating this teleconference, I should explain that Mr. Bazell has been called away to cover an NBC News story. As director of the Choices and Challenges series, it's become my task to fill in for him. Actually, it's a very pleasant task because we have a superb group of presenters, and I think we should get started. Let me introduce them right now. We have Robert Cook-Deegan, physician at the Institute of Medicine of the National Academy of Sciences. Norton Zinder, molecular geneticist at the Rockefeller University. Martin Rechsteiner, biochemist at the University of Utah School of Medicine. J. Robert Nelson of the Institute of Religion, the Texas Medical Center. Abby Myers, who's executive director of the National Organization for Rare Disorders. Daniel Kevlis, a historian at the California Institute of Technology. Our format is going to be as follows. Each of the presenters will speak for about 10 minutes, talking on a different aspect of the Human Genome Project. At the end of the formal presentations, we'll take a 10-minute break, and then after the break, we'll come back for your questions and comments. The panel first will have a chance to talk with each other, and then we invite your questions and comments. The local audience can speak at the microphone in the auditorium. The distant audiences will please use the 800 number that will appear on the screen from time to time. Your questions will be written down and brought in, and we will share them with the entire audience here. Let's begin. Our first speaker, Robert Cook-Deegan, is currently at the Institute of Medicine of the National Academy of Sciences, where he directs the Division of Biobehavioral Sciences and Mental Disorders. But prior to serving in this position, Dr. Cook-Deegan spent a number of years as senior associate with the Office of Technology Assessment of the U.S. Congress. Part of his work there involved analyzing and evaluating the then proposed Human Genome Project and producing a major report for Congress about the wisdom of embarking on such a scientific enterprise. He's just recently authored a book, Gene Quest, that deals with the science and politics of the Human Genome Project. Dr. Cook-Deegan has thus had the unique opportunity to study the evolution of our current effort to map and sequence human genes from its inception to the form it exists in today. He'll be providing us now with a historical introduction to the Human Genome Project. Dr. Cook-Deegan.
I would like basically to answer two questions in the next 10 minutes. The first is, what is the Human Genome Project? And the second question is, where did it come from? The process I'll use to go through that is to first try to lay out what the scientific idea is, and then I'll go through the process of an evolving, the construction of a bureaucratic edifice to support the implementation of that idea. So I'll first talk about science, and then I'll talk about science policy and how the Genome Project came to be part of the federal bureaucracy. The first graphic shows a kind of a cartoon idea of capturing the idea of what the Human Genome Project is. Along the left column, you'll see something of trying to do a map. And suppose you wanted to find the address for every human being on the planet. That's roughly analogous in scale to what we're talking about in doing the Human Genome Project. The number of people on the planet, about 5.5 billion, is roughly equivalent to the same number of A's, G's, T's, and C's that Doris talked about in the introduction. That is, the constituents of human DNA. Every cell in the human body has about 6 billion nucleotides inside of it. That's the code that is translated into the molecules of life. Now, the idea of a map, why would you want a map? And I think the easiest way to explain that is to give you a part of my personal interest in the Human Genome Project. It dates back to 1976, when a friend of mine was diagnosed with Alzheimer's disease. Alzheimer's disease, you know, we don't know what the cause is, and we wanted, as scientists, to try to understand what causes Alzheimer's disease. One way of trying to approach that problem is to find a family where Alzheimer's disease appears to be a genetic trait. And what that suggests is that Alzheimer's disease in certain families is caused by a gene. And for the purpose of the discussion, it's a simplification, but for the purposes of thinking about it right now, just think of a gene as a string of DNA, a piece of DNA, the A's, G's, T's, and C's that encode something happening in a cell. Now the question is, what causes Alzheimer's disease, and what is the gene that causes Alzheimer's disease? You start with a family, and then you say, where is that gene? How can I locate it? What you need to be able to locate a gene is a map of the territory. And the territory is shown in this first graphic, the second graphic. This is basically all of the human chromosomes. All of the DNA is stacked into 23 pairs of chromosomes in each cell. And basically, what you want to do is to be able to find which of those little pieces of DNA has the gene for Alzheimer's disease. Suppose you could somehow spray paint those chromosomes so you could tell which piece of which chromosome was inherited from your mother and which piece was inherited from your father. If you could do that in yourself and all the people going back, that basically means you could trace the inheritance of pieces of DNA through the chromosomes and through families. So in the case of an Alzheimer's family, you could figure out which piece of DNA was always inherited with Alzheimer's disease in that family. Now, so what you want is a map of the territory that allows you to locate the gene. The process for trying to find a gene is called genetic linkage. The bridge from the physical process of going from the inheritance of a character in a family, that is a trait, and trying to find its location is called genetic linkage. And I don't want to go through in elaborate detail what this slide represents, but basically the idea is if you get one copy of a gene from your mother and one from your father, you need to be able to tell those two pieces of DNA apart. You need to be able to tell which piece of which chromosome was inherited from the mother and which from the father. And you go through bit by bit all the way through the genome, and you look for, you always inherit this piece of chromosome 21 along with Alzheimer's disease in this family that has Alzheimer's disease. That process is called genetic linkage. The technology for spray painting the chromosomes, being able to tell them apart using genetic linkage, was developed in other organisms in the 1970s and was first thought about application to humans in April of 1978 in Alta, Utah. So suppose that you can suddenly, you've located somewhere on chromosome 21, in a little piece of it, there is a gene that causes Alzheimer's disease in these families. What you then need to do is to be able to go right to the DNA itself and try to find its constituents. So how do you do that? What you need is a lot of pieces of DNA. So you take the native DNA from the chromosomes, and we've lost the slide in this room, but it'll show up in the graphic, I hope, for the TV audience. Basically, you break a chromosome into small fragments. You make lots of copies of those fragments, and then you find markers that on the graphic for those in the TV audience, you see the red, green, yellow, and orange. Basically, you've taken all these fragments, you've made lots of copies of them, and you have markers that you can tell what their order is. You put the fragments back in order by finding those markers on the pieces of DNA. And so you say a gene is in this part of chromosome 21. You have a way of going back into a test tube in a freezer somewhere and saying, I know that in this test tube contains the DNA that encodes the gene for Alzheimer's disease. And then you start studying the DNA itself to try to find out what it is that's causing it. Now, in some part of the cascade of events of getting down to the Alzheimer's gene itself, you need to decode the DNA code. That is, you need to read out the sequence of A's, G's, T's, and C's. And you look for a correlation between a spelling error in DNA and the inheritance of Alzheimer's disease. So when all that's done, you've got a way, a map, basically, for finding the location of a gene, grabbing the DNA, and decoding the DNA. And that's what the Human Genome Project is. Now, along the way to doing that, you need a set of tools. You need a new set of technologies. You need new biological methods, new instruments, and a lot of computer power to do the analysis that allows you to trace the inheritance of pieces of chromosomes through families. That's what the Human Genome Project is. It's maps. Genetic linkage maps, physical maps, and DNA sequence information to make sense of it. Now, the history I've given you is kind of what you would go through if you wanted to find the inheritance of a disease in a family. That's actually the backwards of how it evolved historically. 3 people in 1985 independently had the idea of, why don't we take all of the DNA in human cells and translate that, just completely decode it. Let's take the sequence of all those A's, G's, T's, and C's and lay it out for all of the human chromosomes once and for all. The first person to do that was Robert Sinsheimer in 1985. He was chancellor at the University of California at Santa Cruz. He had a meeting and he said, why don't we think big in biology? What do you guys think? He brought a bunch of experts together and said, what do you guys think about this idea of sequencing the human genome? The people invited to that meeting thought he was crazy at first. By the time the meeting was over, they said, well, maybe we could do it. Maybe the technologies are mature enough to think about this seriously. Independently of that, Renato Dubecco, whose main interest in those days was human cancer, thought, well, the only way that we're ever going to really understand what's happening in cancer is to lay out all the genetic information in our cells and have a research tool for being able to tell what it is that's causing cancer. What is it that changes in the chromosomes to cause cancer? The only way we're going to do that is to lay out the whole DNA code. So he published that idea in early 1986. He began giving lectures on that topic on Columbus Day of 1985, completely independently of Sinsheimer's idea. But neither of those two ideas really got translated into a program. The place where that happened was in the head of Charles DeLisi. Charles DeLisi worked at the Department of Energy. He'd worked there for a couple of months in 1985. And he struck on the idea of, well, we've got all these fancy lasers, we've got a lot of computers within the Department of Energy, and we care about human mutations. We're trying to study human mutations. Why don't we lay out the whole DNA code, sequence the genome, and then we'll have a research tool for understanding these processes. He had control of about a $300 million a year budget for doing biology within the Department of Energy. And he said, why don't we sequester about $4 or $5 million next year and start a program to develop the technologies and begin this process of melding computers and molecular biology together and new technologies and instruments and eventually have as the goal sequencing the whole human genome. That translation of an idea into policy first took place in Charles DeLisi's head. He actually cordoned off a piece of the budget for the fiscal year 1987. And as that was going on, his idea began to percolate into mainstream biomedical research. The Department of Energy is a little piece of a very large enterprise. It's about 1/40th the size of the NIH research budget for biomedical research. Now, when it came into the mainstream, the geneticists and molecular biologists looked at this idea and thought it was crazy. They said, why would we want to sequence the human genome? And that kind of initiated a process that went from December of 1986 until sometime in 1988, when the idea of the Human Genome Project shifted from having DNA sequencing as its primary goal, with mapping and technology development as subsidiary goals along the way. Those got reversed. And what became primary was the idea of constructing genetic linkage maps, physical maps, and doing enough DNA sequencing to make sense of those things. So DNA sequencing went from being the primary goal to being the subsidiary goal to generating maps of the human chromosomes. In essence, what we saw was a collision of three forces, really. New technologies for analyzing DNA, a tradition of human genetics for trying to hunt down disease-causing genes, and a way of doing molecular biology that grew up in the post-war era and is really the analysis of DNA structure and the technologies that go along with that. During that process of redefinition, the bureaucratic edifice grew. The Department of Energy program got a little bit bigger. And the NIH, the National Institutes of Health, took a look at this little kid who said, I've got this big project, I want to carry it out. The folks at NIH, embodied by the director, James Weingarten, took a look at this and said, hey, that's our business, that's not your business. We should have a piece of this pie, and we should be running the action. We should be running the game. So that also, while the project was being redefined, there was also a jockeying back and forth between the National Institutes of Health and the Department of Energy. And over that course of 2 years, this initial competition was transformed into an alliance. Beginning in 1989, essentially, there were two programs running in parallel in two different departments in the federal government, getting along fairly well, doing some joint planning where it was necessary. And the Genome Project went from being an idea to being a science policy question and a stream of funds for doing certain kinds of biomedical research, specifically that aimed at constructing these maps and developing those technologies. One other conspicuous event in 1989 is that James Watson, the co-discoverer of the double helical structure of DNA, perhaps one of the most famous scientists in the world, became the director, was appointed director of the Human Genome Office at the National Institutes of Health. And at a stroke, the project, which had already been the hottest story in biology, suddenly the program was perhaps the most conspicuous scientific program in the constellation of federal science programs. Now, beginning in 1990, there was kind of a backlash. There was a sense that, well, there's an awful lot of new money going into this genome stuff. And by dint of history, just as the Genome Project was growing by leaps and bounds, there was a squeeze on the rest of the National Institutes of Health budget, and in fact in science budgets more generally. The rate of growth was much slower in other parts of research, and it got harder to get new grants. So while this kid over here was growing really fast and getting a lot of nutrition, the rest of science was feeling starved. And there was a funding backlash, and we'll be hearing a little bit about that. The Genome Project, the rates of growth began to decrease a little bit beginning then, and it really became a part of kind of normal science. Its particular function was constructing these maps. Now, one other distinctive thing that will come up, I suspect, as we discuss things this afternoon, one other distinctive element is the changing contract between biomedical research and society at large. In the post-war era, the biomedical research community was really a bunch of white knights who were out there crusading against diseases of various sorts. And in the 1980s, I think there was a gradual, almost imperceptible shift in public perception of the research enterprise. It got bigger, and the armor on these white knights got a bit tarnished. There were scandals about fraud, misuse of funds, wrong results being reported. And also, the research enterprise grew from being a relatively small and insignificant piece of the federal budget to being one of the larger and juicier fruits out there that could be squeezed. At one point, there was a proposal to take about $12 million of the $60 million for genome research and give it to a program for the homeless. Why? Because the genome budget was big enough and growing fast enough that it was an attractive source of new funding for something else, some other laudable goal. While all that was going on, at the same time, in the post-recombinant DNA era, I think there's a new social contract between science and society. And one of the bureaucratic innovations was the creation of the program that's supporting part of this conference today, which is the Ethical, Legal, and Social Issues Program. It's an unprecedented bureaucratic innovation, really. Here was a commitment made to support research on the impacts of science at the same time as the science itself was going on. Now, why is that? Dr. Kevlis will be going into some of the checkered history, the checkered social history of genetics. So there's a special discomfiture with human genetic information. As we're creating lots of it and putting it out in the world, policymakers were uncomfortable. Aren't some people going to get hurt by the information that we produce in the process of doing all this genome sequencing and mapping? A final observation. The story is actually a fairly clean story. It began in December 1986, and by 1989, we had federal programs in the U.S. federal government, two of them, Department of Energy and National Institutes of Health. And in fact, there were programs being mounted abroad that others will talk about later. An interesting phenomenon, very fast by policy standards. What is it that's distinctive about the Human Genome Project? And I'll just conclude by saying I don't know what the explanation is fully. I think it's partly the power and stature of those who were supporting the Genome Project, and in part it was a sense that policymakers could at root kind of grasp that there was something special about trying to once and for all map what is contained in the DNA in every cell of the human body. That seems inherently like an exciting and interesting thing to do. Thank you.
Norton Zinder, our next speaker, is Professor of Molecular Genetics at the Rockefeller University in New York. Professor Zinder has had an impressive scientific career during which his work has centered on the genetics of bacteria and of the viruses that attack bacterial cells. He's also helped provide direction to scientific work carried out in this country and internationally through his service on many advisory committees and panels. He was one of the scientists who, in 1974, called for a temporary ban on certain types of genetic engineering research until the scientific community itself could examine the risks associated with that research and come up with some suitable guidelines for carrying out this work. It is this particular line of research that has, in fact, led to the very ability today to map and sequence human genes. From 1988 to 1991, he was chairman of the committee that advised the National Institutes of Health about the Human Genome Project, and he is a member of the Council of Founders of HUGO, the international organization that coordinates genome research around the world. Dr. Zinder is exceedingly well qualified to provide us with the scientific goals which are guiding the Human Genome Project. Dr. Zinder.
Bob has already gone through the parts of the Genome Project that I love best. Genetics 101, which is actually what I usually teach, how genes are mapped and moved around and how you do recombinant DNA and things like that. He's touched on that. He's also touched on the history of the Genome Project, which is another rather interesting thing. And, you know, one has the impression that the Genome Project has been going on forever. But actually, I sort of date the beginning of the Genome Project in January of 1989, when there was the first advisory committee, which I chaired, of the Human Genome Project at the National Institutes of Health. So today I'm going not to be either the scientist or much. I'm going to probably sound much more like a bureaucrat, which I'm not, since I'm no longer even associated with the Genome Project. But I want to make it clear, as go over probably much of the ground that Bob already has, what was done to turn 5 years worth of talk about genome into a set of operations which are now functional and fully viable. First, let me say what the Genome Project is not. It is not a project to study all of human nature. It is not a project to study all of human biology. It is not even a project to study all of human genetics. It is a worldwide programmatic effort to improve the technologies for analyzing the human genetic material, for mapping and sequencing it, and to actually do these analyses. Not only do we develop the technology, but we actually do the experiments. For humans, as Bob noted, this means the mapping of some 100 to 300,000 genes located on our 24 chromosomes, and ultimately the sequencing of 3 to 6 billion nucleotides of genetic information. Thus, thereby, there will be provided the infrastructure to influence and even accomplish the understanding of those areas of endeavor that I previously mentioned, human genetics and human biology. I posit that any system that is not willing to devote a few % of its budget to maintain and remodel its infrastructure will suffer the same fate as the city from which I come, New York, crumbling roads and falling down bridges, and so forth. It is clear that we live in the era of the gene and genetics. It is our great technical and theoretical successes of the recent past that have brought us there. Thus, science moves where it can, when it can, and most always will be open to changing procedures in changing circumstances. After all, for years we have lived with the National Institutes of Health, whose institutions were named for diseases, while supporting our basic science, which we said would help to ameliorate the cognate medical and social problems. Some of us even meant it. The Genome Program was designed to efficiently remove some of the drudgery from doing science and thereby move forward more rapidly our efforts to understand the human biological and genetic program. The genome effort, by acquiring the large-scale genetic data, may even change the way our science is done. We now go from the gene to the database and fill in the blanks. Some say in the future, first we'll view the database, then we'll decide which gene or genetic region to look at to those studies, those problems which are of interest and import for human beings. The Genome Initiative has two other important attributes. As we build the infrastructure, each piece adds to the growing whole, so that the more one knows, the more one knows, with the potential utility increasing in almost exponential fashion in the early and mid times. Thus, information starts accumulating from the outset, and already has to a great degree. Secondly, in a real sense, after all, the whole of the data has to be only collected once. There is only one human genome, in a sense. Comparison can then be made of small specific segments, and thus the costs and the potential benefits of the human genome will be amortized over all of human history. Clearly, we are in a win-win situation. The U.S. Genome Program is jointly managed, as mentioned, by the National Institutes of Health, and the Department of Energy. And we put together a program plan 2 years ago. Can I have the screen? What's that? The first item on this plan is to construct a high-resolution genetic map of the human genome and find all of the genes, find all of the 100,000 genes. And by a high-resolution genetic map, I mean a map at about 1% recombination. Simultaneously, we should produce a physical map. As Bob said, we somewhat shifted from sequencing to mapping. To produce a physical map of all of the human chromosomes and of the DNA from certain model organisms. These model organisms are the bacterium Escherichia coli, yeast, the worm Caenorhabdus elegans, and the geneticist's favorite organism, Drosophila, the mouse, and the human. These organisms will be emphasized in order that the most important parts of the evolutionary events can be ascertained by comparing our genetic information. The next is to complete the sequence of the human DNA and the DNA from the model organisms. And this is a contingent plan. We do not intend to go into human DNA sequencing in a production mode until such time as the technologies have been developed to reduce the costs to well below 50 cents a DNA base pair. And of course, we must develop the capabilities to collect and store and distribute and analyze this large amount of the genetic information. More than 100,000 human genes, their localization on 24 chromosomes, and in the end, 3 billion bits of sequence data. In addition, we have to create the technologies to do these things. Monies are being put in to develop robotics for handling of DNA, for developing new technologies for doing sequencing to make it much cheaper. I do believe that ultimately one will be able to sequence DNA at probably a penny a base pair, and that DNA sequencing will become a diagnostic in your doctor's office. And lastly, as Bob mentioned, create an ongoing program in ethics, legal, and social implications of the program. We were well aware that genetics has a checkered history with regard to the way it's viewed by mankind, and that it was necessary to absolutely build into the program, not as an ancillary, or try to convince the NIH or the DOE to have programs in biomedical ethics. There is only one program currently extant in biomedical ethics, and that is built into the Human Genome Program, and it is appropriated with 5% of the budgets of both the NIH and the DOE. I illustrate one of the programs, actually should two, because I believe this program itself is supported by ELSI, but one of the more important things that the Genome ELSI program, and no one else would touch it, was doing the pilot screening for the cystic fibrosis when the cystic fibrosis gene was isolated. And I can assure you, there have been many studies already and thoughts already what would happen if tomorrow a cancer gene, for example, human breast cancer, were isolated. Now let me just say few more things. The Human Genome Program is not an American program. It is an international program. Countries and regions have all produced programs that are appropriate to their area. In some places, it may be no more than an upgrading of their human genetics. In others, it is a quite serious program, and I intend to say a few words about one of them. But there are now programs in Australia, Canada, Latin America, Chile, Denmark, the European Community, France, Germany, Italy, Israel, Japan, Netherlands, Sweden, United Kingdom, and what we used to call the USSR, or now Russia. I would estimate that at the current time, there is something of the order of $300 million being spent on the Human Genome Program worldwide annually. Unfortunately, there's still the area is still sufficiently open that there is very little evidence of detailed competition. Now, always talk about the American program and what we're doing. I want to say a few words about the French program, a part of the French program, because it's rather unique. A part of the French program is called Genethion, and it is supported by money from the Muscular Dystrophy Society. In fact, it's probably the French equivalent of the Jerry Lewis Telethon. It has an institute. It has some 150 people who worked there. They have recently, now I'm going to get a little technical, they have recently succeeded in producing what we call YACs, that is yeast artificial chromosomes, a way to break down that large amount of DNA into pieces that we can handle more or less in the laboratory, into 1 million instead of 3 billion nucleotides, what we have, 0.3%, 1 megabase or 1 million base YACs have been efficiently produced. But more importantly, because lots of people have been able to do this, is that they seem to be rather clean. Two, that they have been able to identify these YACs to particular chromosomes. That means there would be approximately 30,000 YACs for a whole genome, but for one chromosome to be something like 1/10 of that. So 3,000, you're down to trying to find out how 3,000, and that's a difficult problem. But there are techniques for doing this. There are techniques that we can conceive of doing it. They are very difficult. They involved techniques which we call fingerprinting, and the fingerprints were so complicated that they were very difficult to analyze. As I understand, on the basis of new software, which I even hear may have been developed by some Russian immigrants that they managed to pick up because of the events in the Soviet Union, they are now capable of ordering the YACs so that you can tell which YAC follows any other YAC. So it looks like a very important contribution has been made, may have been made by the French to the physical mapping. And once the physical mapping is done, the genetic mapping will be quite easy. I want to close with the following prediction. This is, again, based on our goals, that in 15 years, we will have a 3-inch compact disc upon which will be written the 100,000 or more genes of the human genome and the 3 billion nucleotides of the human genome. Thank you.
Our first two speakers have given us a good foundation for understanding the origin and the direction of the Human Genome Project. Our next speakers will be enriching our appreciation of the range of issues that have been or will be raised by this scientific program. They'll be giving us some additional scientific, ethical, social, and public policy perspectives. First off, we'll hear about some reservations about the project that have been voiced from within the scientific community. To give us this alternative scientific view, we have as our next speaker Martin Rechsteiner. Dr. Rechsteiner is professor of both biology and biochemistry at the University of Utah School of Medicine, where he has conducted his research for more than 2 decades. His research interests during this time have centered on cell proteins, in particular how proteins are broken down and recycled in very precise and organized ways within cells. Among his many important research findings, Dr. Rechsteiner has been able to identify certain regions within proteins called pest regions, for the 4 letters representing the amino acids they contain. These pest regions seem to be the signals that stimulate the process of protein breakdown. Whether Dr. Rechsteiner himself may be serving an analogous function as regards the Human Genome Project, we'll have the opportunity to judge now for ourselves. Dr. Rechsteiner.
Thank you very much, Julie. I really want to thank the organizers for the opportunity to present an alternate view to the Human Genome Project, because it's gotten so many favorable reviews in magazines varying from Business Week to the Smithsonian, and this is a nice opportunity to present an alternate view. The Human Genome Project raises a number of important questions. These encompass ethical aspects surrounding the use of the genetic information derived from the project, whether scientists or politicians should set scientific policy, whether big science or small science is a more productive endeavor, and to what extent media hype should play a role in the setting of science policy. I am a critic of the project, and my criticism focuses mainly on the issue of public policy. In this regard, I have 5 criticisms. First, I believe it is, frankly, a pork barrel project, right? Second, I believe much of the information to be derived from the project is simply not needed, and I'll go into that in more detail. Third, I believe it's a divisive project. It's caused a lot of rancor within the scientific community, and even today within the members of the HGP group itself, there's divisiveness. Fourth, I believe that it is a poor training mechanism. It does not teach young scientists how to think critically, but it's more an engineering project. And finally, I think it's an egregious example of hyped science. All right? I'd like to give my history, which is a very short history compared to Bob's, and that is the HGP has unique political origins. The project did not arise from a broad consensus among scientists or physicians that the information was needed. Rather, it was, in fact, its origins were at, in fact, the Los Alamos laboratories in New Mexico, and Charles DeLisi was a prime mover. And in fact, Senator Domenici from New Mexico was a prime mover in getting the project going, introduced a bill that made special funds for the project. So in this regard, it's got a real special history, at least for an undertaking in the biological sciences. Now, who should formulate science policy? I don't believe that scientists have an inherent right to federal monies, all right? Every society has to decide how much of its wealth is devoted to research, technology. I suppose wise societies invest a little more, and not so wise societies invest a little less. But once that decision has been made, there's no doubt that choosing the projects from among the myriad projects available should be based on scientific and not political criteria. All right? Prior to the HGP initiative, the NIH was already spending $300 billion a year to study and map human genes. All right? Prior to the HGP initiative, the genes for cystic fibrosis had been mapped, muscular dystrophy, and numerous cancer-causing genes had been mapped by traditional scientific endeavors funded by traditional mechanisms from NIH. Indeed, the NIH, the addition of the Genome Project brings the amount of money for this endeavor from $300 million per year to a staggering $500 million per year. And this money, the extra money, was added, I believe, principally through the efforts of Senator Domenici. In effect, we have a senator from New Mexico who is setting scientific priorities. That would be my contention. Now, will the HGP provide valuable information? Right? Good biological approaches might be defined by the insight gained relative to the effort expended. Using this criterion, the HGP is certainly a questionable endeavor. Consider, for example, how useful is a cDNA sequence, the very object of this massive undertaking, right? The sequence for the important human oncogene, P53, has been known for a decade. This is a gene that is mutated in perhaps 40 to 50% of all human cancers. We've known its sequence for a decade. We still have no understanding of what the protein does, and the knowledge of that sequence has not advanced any therapies based on that oncogene. All right? All right? I contend that sequencing a disease-causing gene will provide medical insight only in proportion to how much basic physiology or biochemistry is already understood. New cDNA sequences will often be of little value unless there's a surrounding physiological database with which to interpret these DNA sequences. Right? And as for the 95% of the DNA in the genome that most believe is junk DNA or pseudogenes or repetitive sequences, I believe these serve mainly to separate out the genes that encode these bodies, all right? And in fact, knowing the sequences of these pieces of DNA is a wasted effort, by and large. Right? Now, the HGP, the Human Genome Project, is poorly formulated policy for other reasons. It has created a deep rift within the scientific community. all right? At a time when less than 1 grant in 8 was being funded, right? When the grants that were being funded were being cut by 25%, when university labs were being starved, as we've heard, at that time, we saw a proposal to invest hundreds of millions of dollars in a small number of scientists. And in fact, in 1990, 10 individual scientists received close to $20 million per year annual funding. That's almost $2 million annual funding per year, when an average NIH budget for an investigator in a university is about $100,000 to $150,000 a year. So we were talking about grants that were 20 times the typical NIH grant, going to a very small number of individuals. All right? Awarding mega grants to a small number of individuals, while investigators such as Trina Schroer, shown on the slide here, could not get funded, a talented young investigator, was bound to cause resentment, all right? And a number of us, myself included, were angry that talented young scientists were not being funded, while well-established older scientists, already well-funded, were receiving even larger amounts of money, all right? This did not seem to be a very good public policy, all right? Okay. Even today, the divisiveness between big science and small science has extended to the Genome Project, where there are patent fights. So the Genome patent fight erupts. That's shown on the news and comment on the screen there. And in fact, apparently has led to some dire consequences within the Genome organization itself. All right? So one can also criticize the project for its role in training young scientists. The HGP diverts funds from university laboratories to centers. And in this regard, it impacts negatively on the training of the future generation of scientists. All right? $200 million, the proposed annual budget, translates into about 1,000 grants to universities. And I calculate that there are about 150 major research universities in the United States, so that's about 7 grants per university. If you take that 1,000 grants and put them into the university setting, they train undergraduates. The monies provided train graduate students, undergraduates, postdoctoral fellows. If you take the same amount of money and put it into a few centers, you end up training mostly technicians, all right? Right? In fact, Walter Gilbert, who's an advocate and a recipient of a grant from the Genome Project, claims that the endeavor is not suitable for the training of postdoctoral and graduate fellows, and in fact is using production line, his term, production line technicians for the task. So we have here a public policy that diverts large amounts of monies from universities to centers. I claim in the long run will divert money from the training of graduate and postdoctoral fellows to the production of product, of which already I'm not so enthralled. I'm not enthralled by the 95%, knowing the sequences of 95% of the genome, even if it's obtainable on 3 inches of disk, all right? All right. It's simply not useful information in my mind, all right? And now I would like to close by touching on one last aspect of the HGP. Defense of this initiative has resulted in some of the most outlandish hype in recent memory. And I'm going to quote from Paul Billings, who actually works in the Human Genome Center in Berkeley and is a supporter of the project. And he has stated in a letter to Science, advertising the Genome Project with slogans equating it to the genetic equivalent of the Holy Grail, the essence of human life, in quotes, a blueprint for understanding human biology, creates an aura of unbelievability around the project that's bound to lead to a public political backlash. I firmly agree with that. All right? I think all of us would agree that all citizens, scientists included, owe it to their society to consider the benefits of their activities to that society. And also, I believe that all scientists have a responsibility to present reasonable, absolutely reasonable estimates of the success or failure of their projects and the utility of their projects. And I believe one could make a strong case that in the case of the Human Genome Project, an excess amount of hype has been used to sell this project first to Congress, then to the public, and perhaps not so well within the scientific community itself, all right? But also within the scientific community. So in summary, I believe the HGP represents a series of bad policy decisions. It was initiated for political, not scientific reasons, all right? It offers little opportunity, or certainly less opportunity, to train the next generation of scientists, right? It's generated deep division within the scientific community, all right? And the genome projects, of which there are now many, in fact, should be curtailed. The research funds liberated could be used much more wisely in the quest to solve human diseases.
It was clear to everyone from the start of the decisions about a human genome project that a human genome project of any kind would be certain to raise a number of ethical and religious questions. Helping us now to identify what these ethical and religious aspects are is J. Robert Nelson. Dr. Nelson is director of the Institute of Religion at the Texas Medical Center in Houston. He has worked, and indeed continues to work, in the international religious community, having served for the World Council of Churches in Geneva, on the National Commission for UNESCO, and with the European Society of Culture in Venice. And he was the first non-Catholic to teach at the Jesuits' Pontifical Gregorian University in Rome. Dr. Nelson has delved deeply into the ethical and religious side of genetic engineering, co-authoring the report on genetic engineering from the National Council of Churches, consulting for the President's Commission here in the United States, and authoring a book entitled Human Life, A Biblical Perspective for Bioethics. Most recently, he has organized a multi-year study on genetics, religion, and ethics, which is focusing its attention on the Human Genome Project. I note that Dr. Nelson was named to the Sports Illustrated Silver Anniversary All-American Football Team, giving him just the skills he needs to lead us through some difficult and potentially risky terrain. Dr. Nelson.
Thank you, Doris. We have a matter of reciprocity here because only 2 weeks ago, Professor Doris Zallen was attending our conference on genetics, religion, and ethics in Houston, and now I have a chance to show my respects to Blacksburg by coming here. I was asked by my wife, why are you going to a place like Blacksburg? I said, well, I'm going to join a lot of VIPs at VPI. But don't count me as one of the VIPs. Speaking of alphabets, we've heard about ELSI, E-L-S-I, as the acronym for ethical, legal, and social issues in the Human Genome Project. I would suggest that the letter R ought to be added to those because the religious factor is one which has been recognized from the very outset of this enterprise and which becomes for many equally important in many ways, as I'll try to show. Already, in fact, in the United States, some 6 national denominations have already fashioned their policy statements with regards to genetics and genetic engineering, and a very large number of conferences and studies are taking place under this problem. So our religion and ethical behavior are being influenced by genetics very much as other aspects of our life. And the data and the knowledge are being produced in such a way by the Human Genome Project and by all other kind of genetic research that the horizons of our understanding of human life are being broadened in a most immeasurable way. First, for many who believe in the divine creation of the world, the genetic data simply enhance the sense of the wonder of creation. Never before our time have people been able to know so much about the astounding structure and performance of the DNA molecule, of chromosomes, and of cells. We, in fact, are the first generation in all of human history who have been able to learn of this. Now, ever since ancient times, people have wondered about creation. The psalmist in the 8th Psalm says, when I consider the heavens and the stars and the moon, what is man, what is humanity in comparison to them? Or another psalmist says, we are fearfully and wonderfully made, even in our mother's womb. Or in the 18th century, there was a great sense of wonder about the world. The great poems set to music by Haydn on the spacious firmament on high, the creation, ends with the words, the hand that made us is divine. In other words, by seeing the immensity of space and recognizing the great complexity of creation itself, there is an indication of the divine creator's hand. Well, that was precipitated very largely by astronomy. But now today we are going not to the infinite spaces beyond, but to the infinite spaces within and beneath us. That is to the almost infinitely small measurements in terms of space and time. And I'm constantly amazed as I talk with my fellow workers in the fields of genetics and science and learn about what they're doing to realize that they are manipulating invisible quantities virtually. Almost unable to see the very things which are so instrumental in the fashioning of our lives and often, too, of our behavior. But not only the small size, but the prodigious functioning of the express gene, making proteins and determining the tissues and organs and their development and causing diseases, and also, I'm very glad to say, giving the power of healing. All of these secrets of the world, so to speak, in the human and organic life are becoming known to us. Furthermore, we learn in our present time about the wonder of the organic creation itself. The millions, literally millions of species of organic life and animal life in the world, far more than any of our predecessors ever dreamed. And the great thing about that is that we are discovering too, and it's perplexing to some, that the DNA structure in our own cells is shared in common, it is said, by a very high proportion, in some animals up to 97% of the same kind of DNA material which we have in the human person. So these are staggering facts, and we have to take account of them. As with all reflective human beings, we try to understand who we are, why we are here, and what this world is about. But secondly, we are compelled to ask, is there any purpose in evolution? This is not to cast doubt on the countless evidences of organic evolution. That is not the purpose here. Biblical religion does not contradict the origin, or for that matter, the extinction of tens of millions of species over the eons of time. Genetic science today magnifies the evolutionary evidence. It is found not only longer in fossils, fossilized bones and shells alone, but in the vast quantity of differentiated cells and the genes of DNA which determine the continuity of living beings as such. Again, a cause for wonder. But where is any meaning in all of this? Where is any purpose found? Do developing changes of shape and character in human beings, for example, testify to any sense or reason or purpose, or are we simply here and that's all? Many agnostic scientists deny any purpose, what is called any teleology, any movement toward a given end. Great scientists like Jacques Monod, Edmund Jacot, Edward Wilson at Harvard, the popular Carl Sagan, many of them are saying that we are simply created at random. We are going no particular place. We don't know where or why, but here we are and we have to make the most of what we have. Even human life itself, they say, often called by others the crown of creation, testifies to the random power of chemistry and physics, and not to any inherent or even divine purpose. And so all of our inquiries into the genetic structure of not only the human being, but also all organic animal life and plant life, all of this is raising for us fresh new questions about the understanding of a purpose in our existence. Meanwhile, other scientists, and very many of them I'd point out, not just a few, other scientists are declaring that with the purpose of human development, we, with our present and growing knowledge of genetic engineering, are actually starting to take control of our evolution, not being chips on the great sea of evolutionary change, but rather those more likely to be steering the ships. For there is a mystery of human nature, is there not? A mystery always eluding satisfying definition. Thoroughgoing scientific materialism would reduce human nature to the countless differentiated genes of one's body, including also of one's brain. The latest report on genetic therapy from a committee of the British Parliament, for example, just as 1992, states this. It says it is the interaction of genes and environment that, quote, confers the quality of humanness. It is genes and environment, they say, which confers the quality we call humanness. That is called biological reductionism. To be sure, our new knowledge of the human genome gives great understanding of how we become the kinds of physical humans that we are, and to some extent also why we behave as we do. But there is much more to life than physical structure and genetic energy, as we learn. On the other hand, the wonders of the genes now dissuade us from thinking the essential human life as being only a spiritual life, which is sort of a retreat from science. To say that simply we cannot understand the functioning and purpose of physical life, therefore the only real life is the spiritual life. That is an ancient idea, both philosophical and religious, which is still alive in various religions and philosophies today. But does it not ignore the reality of our DNA structure, of the genes, of the cellular life which make us up to be what we are? We are not pure spirit. We are matter and spirit. And we are matter and spirit joined together into one kind of organism. Can we resolve the problem of having to choose between a wholly materialistic idea of human nature and a wholly spiritualized one? Indeed, we can, and genetic science helps us here. For the solution is known in the medical profession and in psychology as the psychosomatic unity of the human person. Body and soul, genes and spirit and mind, all of these in a unity constitutive of what we call humanness. And this is incidentally the Hebraic understanding of the Old Testament and of the New on the view of what human life is all about. But moral responsibility has to be considered here too. It is an essential ingredient of being human. Human cultures and civilizations can no more survive without moral responsibility than individuals can. But what accounts for moral responsibility? Do our genes account for it? No, I do not believe so. But genetic research is showing us that some of our moral responsibility, or perhaps our immoral irresponsibility, is affected by genetic endowment and by predispositions, and that's something worthy of exploration. But freedom of the will comes into our consideration here. Freedom of the will, we think, is essential to moral behavior. For example, drunkenness is widely regarded as immoral or at least socially repugnant, and many people do drink too much as a matter of their own choice. But now come studies of identical twins raised in very different environments, but both twins developing early in life the condition of alcoholism. Is that a disease or a voluntary behavior pattern? Is it determined or is it a free choice? This is something which is perplexing, but we are exploring it. Is it a moral question resting upon free will and free choice? Most recent research also has been raising questions in recent months about the origins of homosexuality. Is it a genetically determined state? A matter of choice? How is it? genetic determinism, if it is true, and many doubt that, but if it is true, can explain all kinds of behavior. The leading teacher of sociobiology, Edward O. Wilson of Harvard, the great expert in insect life, really does believe that moral freedom and ethics are an unreal illusion. Even what we call altruism, that is to say, concern for the other, is determined, they say, by our genes. As much in humans as in seagulls or zebras or bees or Wilson's favorite subject, the termites. Religious defenders of free will and moral agency can scoff, if they will, at such notions. But let us be open-minded to possible truth. Even St. Paul of the New Testament, some of you would remember, had this to say about the contradictions in his own life when he said, "The good that I want to do is that which I do not, and the evil I do not want to do is precisely what I do. Oh, wretched man that I am," he says. Wretched man because he lives in this kind of contradiction. So we consider nature and nurture, those two words which look so much alike but which say so much differently. Nurture and nature apparently hold us in a life of tenseness and moral contradiction. Just as the unified duality of body and soul allows us to be one person, so we are both in bondage to our genes and morally capable of surmounting the influences of our genes. The Human Genome Project will continue to adduce more evidence of the power of nature over nurture, or of genes over environment. Likewise, or contrariwise, the mysterious human spirit and intellect will struggle against the genetic inclinations to both disease and to moral evil. Sometimes this contradiction is expressed in the question about man or woman or humanity. Are they a beast or an angel, as René Dubos asks? But in religious, biblical terms, it can better be said that we are creatures of genetic inheritance, but we are made in the image of God. And therefore, choices or chances are always set before us, not only in how we conduct our lives, but also in how we procreate new human beings. A most dominant, far-reaching axiom of religion is the infinite value of each human being. That seems to be a simple statement of incontestable truth, but it is challenged more and more today by increasing knowledge of genetic diseases. Can the same infinite value of life be attached to those destined to be born with most serious mental and physical disabilities? The question of justifying early abortion becomes increasingly acute and vexed as prenatal diagnosis becomes earlier and more accurate in its realities. When the American Society of Human Genetics recently accepted abortion for what they call severe defects, they did not say how severe those defects must be. Or is anyone really justified and required other than a pregnant woman to make that decision? Well, the division of religious belief on abortion, those divisions are as evident and as notorious as in secular society at large. No one has yet been able to announce the perfect solution, and we do not look to genetic science for that solution. Instead, genetic research and reproduction technologies are going to serve to complicate the question even more than it is today. But more than other people in general, religious believers hold in their trust in the divine creation that life must be adhered to basically as a matter of sanctity, as a matter of holiness, as a matter of trust that is given to us not by ourselves but by God. And therein lie many of our perplexities. In the same way, finally, justice, equity, and love are commanding religious concerns in the field of genetics. They may not seem so when you talk about the laboratory techniques, but the implications of all of genetic science for society and human life show that these terms are decisive ones: justice, equity, love. These are not the possession solely of believers, of course. They are shared by many others for differing reasons and of different persuasions. But believers find the source and the power of justice and of equity and love not in good ideas, but rather in the eternal will of the God who created us all. With a new intensity, therefore, issues are raised by genetic science about theories of race and racial superiority, about individual worth, about the integrity of families and of marriage, about access to genetic counseling, about care for the disabled, and for the unwanted persons of our society, and about the great question, the overwhelming question of world population control. Well, when we think of these terms, our heads begin to swim in confusion. How can we possibly contemplate the complexity and the magnitude of them all? And yet that's precisely what we are dealing with when we talk about religious reality. We are talking about the ultimate mysteries of our existence and about the identity and the purpose and the destiny of the human race. The Human Genome Project, then, will not usher in the perfect society, much less the kingdom of God. But even as it presents new problems, it is offering clues to a better way of human health and of human well-being. And that's why we must continue to devote ourselves to the study and the discussion of these genetic issues. Thank you.
Abby Myers, our next speaker, is founder and executive director of NORD, the National Organization for Rare Disorders, a coalition of voluntary agencies representing people afflicted with rare but serious health problems. She's also served on the Human Gene Therapy Subcommittee at the National Institutes of Health, consulting on policy related to carrying out gene therapy to correct gene-caused disorders in humans. She may, however, be better known to many in our audience for her tireless efforts and advocacy that have led to the creation of the Orphan Drug Act. The Orphan Drug Act provides hope and help for those with rare disorders, a community that prior to the act was largely overlooked by the pharmaceutical industry. Through her work with NORD and her numerous other educational and policy outreach activities, Abby Myers has represented the needs and protected the interests of those afflicted with serious health problems. She'll be drawing on this extraordinary experience to help us today understand better some of the human concerns and societal consequences of the Human Genome Project.
Thank you very much. I'm an ordinary person. I'm not a scientist, and I want to make that clear from the start. And I suspect that many of you in the audience are not scientists and may not even be in school for science. So you may be as confused as I am about these words we've heard this morning about pests and yaks and oncogenes and ELSI. It's very hard to understand all of the technical aspects of the Human Genome Project when the one thing that most people are concerned about is, how is this going to affect me? Not only me, but the children that I have, or the elderly parents that I have. What does it mean to the world now and in the future? Well, historically, society, especially since World War II, has been very, very concerned about eugenics. Eugenics evolved at the end of the last century, and there's now a fear about engineering perfect humans and eliminating humans with flaws. And the fact that there are some people always who would like to see no genetic diversity. We see that we saw that in the Nazis in Germany about what the perfect Aryan would be, and there are other societies around the world who feel that way. Individuals, though, should be very concerned that the Human Genome Project threatens confidentiality of medical information, and it provides an opportunity for intense discrimination. During the 1970s, when they had found the genetic material involved in sickle cell disease, the information didn't get out to the public in the right way, and everybody felt that if you had a sickle cell trait, that you had sickle cell disease. And these poor people who had the trait and did not have the disease and were not affected at all by the trait found that they couldn't get health insurance, couldn't get life insurance, couldn't even get jobs. They were denied everything. And it took a massive federal effort to undo the damage that had been done by trying to teach people what the science really meant, and that a person, because he carried the gene, didn't necessarily mean that he would get the disease. So right now, we're in the same position. It's as if we all have the sickle cell trait, and society hasn't come up to the scientific understanding. If we develop some type of a genetic screening test for any number of diseases, let's take cancer. There are 200 different types of cancer. And if they use that test on you, you will never be able to get health insurance. You will never be able to get life insurance. And you can say to yourself, well, I'll get around that because I'll lie about it. When I'm turned down with one company, I'll get it with another. There's a huge computer base up in Massachusetts called the Medical Information Bureau that has all the medical information on all of us. And you just don't know that your promises of confidentiality, that your doctor is not going to tell your neighbors that there may be something in your family, means nothing, because if any insurance company and any clerk at any insurance company could get into that medical information database and find out your medical history. Discrimination is really going to evolve out of categorizing people unfairly. The predictive tests are the worst thing. A test that says you carry the cancer gene doesn't mean you're going to get cancer, but it means you're predisposed to it. And if something in the environment occurs, you may get cancer. We don't know whether the same thing will happen with the Alzheimer's gene and a number of others. But the fact that you have the gene means that the predictive test is going to predict that you'll probably get it, or you might get it, or there's a percentage that you might get it. And that can affect your employment. Why would anybody want to hire you if the insurance company was going to charge more for your health insurance premium than other employees? What about marriage and reproduction? When we hear from people all over the country who write to us because somebody in their family was diagnosed with a rare disease, they often tell us that as soon as they get the diagnosis for the child, there begins an internal struggle in the family with both the husband and the wife and the in-laws all saying, it didn't come from my side of the family. Just think about your own families. Think about what might be there. Could there come a day that a person would say, no, I'm not going to marry you because you just might have somebody with Alzheimer's. Your grandmother may have had it, and maybe you have the gene. Genetic tests are going to have to be voluntary. If you don't want to know, you shouldn't have to take the test. And what about cost containment? As healthcare costs go up, there'll be intense social pressure for more prevention. That's the way the government and insurance companies intend to save money. They keep talking about things like seatbelts. They keep talking about things like smoking cessation. They don't want you to eat red meat. Well, how are we going to prevent genetic diseases? It raises the specter of abortion that most people don't want to talk about or think about. But if you look at the anti-abortion laws that have been passed in several states over the last few years, you'll find that no exception was made for genetic diseases. What about gene therapy? I think everybody, especially the government, believes that it must be used for therapy and not enhancement. But then there's an interpretation of what is a disease and what is a cosmetic change, for instance. We're talking about changing the somatic cells now, but they're trying to develop the technology to change the germ cells, which means a person's egg or sperm would be permanently changed, and it would affect generations to come. Well, there are many opponents to changing germ cells. They say that it would be usurping God's prerogatives, and it's a slippery slope towards enhancement. Before you know it, somebody's going to say, I want my son to be a little taller. Can you change his growth hormone gene? Or, I don't want him to be black. Give him white skin. All of these things could possibly happen if there's no regulation. They also say that you can't change these cells because we don't have consent of future generations. What happens if 3 generations from now they turn back on us and say, why did you do that? I would have rather have had the gene for that characteristic. Now, it's assumed that gene therapy will give us satisfactory treatments. So the opponents to germ line therapy believe that treatment is all we need. We don't really need these types of preventions. But the treatments are likely to cost more than the cure, and cost will be a real major factor. The research problems, I think that they were just referred to. Dr. Nelson just spoke about this problem. There is an intense disagreement in the scientific community about these Manhattan projects, like not only the Genome Project, but AIDS are taking huge chunks of money out of the research community and putting them into places that are politically preferred for one reason or another. The thing that bothers me personally as I look over protocols, and especially informed consent forms as people agree to go into experimental protocols, is the fact that the hospitals, medical schools are expecting patients to pay for the right to go into some type of experiment. That's not the way it used to be. The university used to cover the cost. And whatever wasn't covered by the university was charged to your health insurance company. Well, health insurance companies a few years ago said, we're not going to pay for anything that's experimental. And so what happened is they started charging patients. Now, what does this mean? It really means that there's a new Darwinian law. It's survival of the richest. And then there's the controversy about commercialization. NIH is coming out with a new strategic plan, and in the draft it says that biomedical research should be expanded in order to enhance the nation's economic competitiveness and ensure a continued high return on the public's investment in research. Is that why we're doing research? This is government policy. Why are we doing biomedical research? Is it because of competitiveness? This really sheds light on the government's view of commercialization and what that really means in the long run. The question of patenting gene sequences has come up. It's been very much in the news lately. And the prices of therapies developed with government funds are incredible. We have a new orphan drug that was developed with 30 years at NIH of your taxpayers' money, and the technology was given to a company who charges $350,000 a year for that drug, for a chronic disease. Less than 250 people in the country can afford it. Do we have a right as a society to say whatever is commercialized out of this Genome Project should be accessible to all of the people who paid for the development? It's not the American way to control prices. We also have a problem in this country of saying we have to win the race. It's like the race to the moon. There was no reason to put human beings on the moon. We could have sent robots. Would have been safer and less expensive. But we had to put people up there because we were racing against the Russians. Well, with the Human Genome Project, we're also racing against the world for the United States to discover all of those genes first. Why? We're spending a huge amount of money. Those other countries have thrown in a few dollars. Most of them are just getting started now. Some of the countries will never get started. Why is the American taxpayer subsidizing this project to the extent that it is? It's going to benefit all humanity, and the world should share the costs. I think that we may be able to regulate the researchers. We may be able to make some major changes in that area, but we are never going to regulate practicing doctors. And the practicing doctors transferred all of this technology to the patient's bedside. I read recently that male weightlifters were having silicone gel implants put in, breast implants, because to build up their physique. And I can't be so angry at the weightlifters as I am at the doctors who are doing it. We need new laws and regulations. We need a law on privacy. You all think you have privacy now. You don't. The only privacy you have is within the federal government if you're the subject of research. They can't let your information out, but everybody else can let information about you out. This privacy law should cover all medical information, even if it's not federally funded. On universal health insurance, are we ever going to get it? And if we are, it's going to have worse cost controls in it and more prevention in it than what we have now. We really have to think about that very, very seriously when it comes to genetic diseases. There must be some restraint on the prices of some of these things. Even if we get universal health insurance, we're going to end up with something like the Oregon plan, where they say bone marrow transplants are too expensive. So we're going to let that baby die because we can treat 5 other children with less expensive disease for the cost of that one baby. We're doing that now, and we're making that decision now. And yet we still have states that are saying that they won't allow abortion, but they still won't accept the medical cost of raising that baby once it's born. We need changes in the patent laws. They've got to straighten out this funny business of patenting genes and gene sequences. And we need civil rights laws for genetic discrimination. People with genetic disorders should be protected, a protected class just like women and minorities. And we have to find out how to better transfer the technology that comes out of the Genome Project so that we get the benefits to the patient's bedside faster. We don't want them to be studied for another 10 years. But with all of these problems that I've just mentioned, I want to tell you that I think that we're not ready. Society isn't ready. If mankind is supposed to be the beneficiary of this project, we should make it clear now that the Genome Project must not be a competitive race between nations, and that all mankind will share in the rewards in the spirit of the Salk polio vaccine. However, in the current environment, reality dictates that even Dr. Salk, who is developing a vaccine for AIDS, has patented it. Thank you.
Lurking behind any discussion of human genetics is the specter of eugenics. Under the banner of eugenics, governmental policies have been made in the past, some of them quite dreadful, that have had an enormous impact on people's lives, even have caused them to lose their lives. Our last speaker, Daniel Kevles, is Professor of Humanities at the California Institute of Technology. Professor Kevles is a historian of science and an expert on the history of eugenics and on the effects such policies have had. His award-winning book, In the Name of Eugenics, explores the history and politics of eugenics movements, and is soon to be published, The Code of Codes, extends his analysis to include the Human Genome Project. Professor Kevles will be surveying the historical record for us and giving us a better means of anticipating where the increasingly more powerful tools of human genetics may be leading.
Thank you very much, Professor Zalin, and it's a pleasure to be here at this conference. As Professor Zalin said, and as Mrs. Meyer has already indicated, the shadow of eugenics does hang over any discussion of the social implications of human genetics, but particularly over considerations of the potential impact of the Human Genome Project. What I want to do here this afternoon is two things. First, to review for you very briefly the history of eugenics, particularly in this country, but not limited to that, and then to turn to our own day and to examine the Human Genome Project's social and ethical implications against the backdrop of the history of eugenics and the apprehensions raised by that history that continue to be with us. First, the history then. Eugenics was a movement that flourished during the first third or so of this century in the United States, Europe, and elsewhere, including Oriental nations, Latin American nations, and so on. It's not just a white Anglo-Saxon Protestant movement. The aim of eugenics was to interfere with human reproduction, to get rid of people who were taken to be biologically undesirable, so to speak, by virtue of their genes, and multiplying people who were judged biologically desirable by the same measure. Eugenicists found evidence that there were many undesirables. They attributed alcoholism, prostitution, criminality, poverty, and mental deficiency, which in those days they called feeble-mindedness, primarily to biological causes, and after the rediscovery of Mendel's laws of heredity in 1900, they attributed these social pathologies and individual pathologies to genes. Now, most eugenic theorizing was ultimately found to be worthless scientifically. Eugenicists tended to neglect that many traits are the product of multiple genes rather than single ones. They also paid far too little attention to cultural, economic, and other environmental influences in their accounts of mental behavior, of mental abilities, such as, for example, low scores on IQ tests, and also to such social behaviors as prostitution and criminality. Class and race prejudice were also pervasive in eugenic science. Eugenicists belittled people who earned lower incomes, and they applied disparaging stereotypes of a biological nature to people who were not of Northern European and largely Protestant background. For example, to Poles, Jews, Irish, Italians, Greeks, and blacks. What eugenicists wanted to do programmatically on the basis of these misbegotten theories of theirs was to increase the frequency of socially good genes in the population, which they called positive eugenics. But they did not know very well how to do that, so they focused on the alternative, which was known as negative eugenics, and meant operationally attempting to decrease the frequency of allegedly bad genes in the population. In the United States, the decrease was attempted to be accomplished by restricting immigration, for example, from southeastern Europe, because all of these people, Italians, Poles, Hungarians, and so on, were alleged to be bringing in bad germplasm to the United States. They also sought to decrease the frequency of bad genes in the population by sterilizing people who were judged to be biologically unfit, so that these people could not pass on their allegedly bad genes to future generations. Indeed, by the late 1920s, some 24 American states had enacted eugenic sterilization laws, and such laws were declared constitutional in the 1927 U.S. Supreme Court decision of Buck versus Bell, in which Justice Oliver Wendell Holmes, upholding the Virginia sterilization statute, said in a famous, or I should say infamous phrase, 3 generations of imbeciles are enough. My own state of California sterilized more people under the eugenic sterilization laws than did all other states of the Union combined. But their total of eugenic sterilizations paled in comparison to what the Nazis did in Germany after 1933. In Nazi Germany, eugenicists celebrated the creation of a super race, and the eugenics movement prompted the sterilization of several 100,000 people, and also, of course, inevitably, ultimately led through the euthanasia movement to the gas chambers and the death camps. Hence, it's no surprise that the specter of eugenics should hang over any discussion of the acquisition of human genetic information and its uses of any type. However, to turn back to our own day and that specter, I consider it unlikely that advances in genetic knowledge will lead to a revival of attempts to produce a super race. While it's true that the Human Genome Project will undoubtedly accelerate the identification of genes for physical and medical traits, it is very unlikely to reveal with any speed, if ever, how genes contribute exactly to the formation of those qualities such as talent, behavior, personality, athletic ability that the world so much admires and craves to have more of. However, the steady increase in the identification of negative genes might well tempt governments and other agencies to pursue a program of negative eugenics. Indeed, there was a proposal to create a Human Genome Project in the European community, which has been touched upon by Norton Zinder here. The original proposal, advanced in 1988, justified the initiative in terms of what was called preventive medicine, that is, or predictive medicine, I'm sorry, protecting individuals from the kinds of illnesses to which they are genetically most vulnerable and to preventing the transmission of the genetic susceptibilities to the next generation. This was all declared in the programmatic purpose of the Human Genome Project as it was first advanced in the European community. Now, economic costs of healthcare figured in the justification in Europe, and it is true that economics may well prove to be a powerful incentive to a new negative eugenics. Undoubtedly, concern for financial costs played a role in the early eugenics movement here and elsewhere. At the Sesquicentennial Exposition in Philadelphia, which you can see up here and on your screens, in 1926, the American Eugenics Society had an exhibit. This exhibit included, as you can see in sort of the upper middle, a board that in the manner of the population counters of a later day revealed with flashing lights, and here's a close-up of this, revealed with flashing lights that every 15 seconds, $100 of your money went for the care of persons with bad heredity, and that every 48 seconds, a mentally deficient person was born in the United States. Thus, it was reasoned, eliminate bad genes from the gene pool, and you would reduce what are nowadays called state and local welfare costs by reducing public expenditures for what was then called feeble-mindedness, in at least its public institutional settings. That is, state institutions and state hospitals for the mentally deficient and physically disabled or diseased, which in every state where there were eugenic sterilization laws were the only places that these laws reached. You were exempt from them if you were in private care or in the care of your family. In our own day, the more that health care in the United States becomes a public responsibility, payable through the tax system, and the more expensive this care becomes, the greater the possibility that taxpayers will rebel against paying for the care of those whom genetics dooms to severe disease or disability. I think the issue here is, I part from Ms. Meyer here in the sense that it's not just the government as some faceless entity which may want to fiddle with the healthcare system and use genetics to reduce costs. It's because taxpayers are paying for what the government pays in the form of health costs. It is possible, indeed, that on grounds of cost, even a national health system might seek to discriminate between or among patients using the criterion of how expensive their therapy and care might be. Public policy might well feel pressure to encourage or even to compel people not to bring genetically affected children into the world, not for the sake of eugenics, that is, for the sake of the race or the gene pool, as eugenicists might have put it, but simply in the interest of keeping public health costs down, not because the government, as some faceless entity, is interested in that, but because the taxpayers who support the government programs are interested in doing that. All of this said, and here is where I part, I think, most sharply from Ms. Meyer, all of this said, it seems to me, however, that a number of factors are likely to offset a revival of negative eugenic policies, at least in the political democracies. Democratic societies now contain very powerful anti-eugenic constituencies. For one thing, awareness of the barbarities and cruelties of state-sponsored eugenics in the past has tended to set most geneticists and the public at large against such programs. Then, too, Handicapped or diseased persons are politically empowered, as are minority groups, to a degree that they were not at all empowered in the early 20th century, at least in the United States. To be sure, they may not be sufficiently empowered to counter all quasi-eugenic threats to themselves, but they are politically positioned and with allies now in the media, the medical profession, and elsewhere, including especially, I might say, the Roman Catholic Church, which has been a staunch opponent of the eugenics since at least 1930. They are so empowered with such allies to block or at least to hinder eugenic proposals that might affect them. Indeed, an anti-eugenic coalition rose up in the European community and defeated the European Commission's Human Genome Project in its original form and translated it into a purely scientific program with a mandatory commitment to research in ethical and social issues related to genetics, similar to the ELSI program here in the Human Genome Project. In sum, then, in my view, the near-term ethical challenges of the Human Genome Project lie not in eugenics, but in the grit of what the project will produce in abundance, that is, in genetic information. The challenges they center on the control, diffusion, and use of that information within the context of a market economy, and in that context they are rather disturbing. The advance of human genetics and biotechnology has created the capacity for a kind of homemade eugenics, where individual families may decide what kind of kids they want to have. At the moment, the kinds they can decide to have or select are those simply without certain disabilities or diseases, such as Down syndrome or Tay-Sachs. Most parents, I think nowadays, Barbara Walters to the contrary notwithstanding, would probably prefer just a healthy baby, not a super baby. They would prefer such a baby if indeed they are inclined to choose at all, which many are not so inclined to do. But in the future, some might have the opportunity, for example, via genetic analysis of embryos, to have improved babies, children who are likely to be more intelligent or more athletic or better looking, whatever those comparative terms may mean. You take your choice. Indeed, a journal called Trends in Biotechnology in 1989 published an editorial that recognized a major source of this pressure on parents. Human improvement is a fact of life, not because of the state eugenics committee, but because of consumer demand. How, the journal asked, can we expect to deal responsibly with human genetic information in such a culture? And we may ask, how indeed? The increasing availability of human genetic information can cut many ways. It can crush individuals by telling them that their child will be diseased, or it can liberate them, and let us not forget that side of it. It can liberate them by assuring them that it will be born normal, undiseased. Genetic information concerning behavioral or personality traits may be reported as reliable by scientists in press conferences and then later proved wrong, but no press conference. This report can very well stigmatize people in the interim or forever. Employers, as Ms. Meyer has suggested, may seek to deny jobs to applicants with a susceptibility or an alleged susceptibility, and so far it's mostly alleged, to disorders such as manic depression or illnesses arising from features of the workplace. Life and medical insurance companies may well wish to know the genomic signatures of their clients, their profile of risk for disease and death. But the response to that is not an automatic blanket insistence upon privacy, because you can have asymmetry of information where a prospective client knows that he or she is going to get sick because he or she knows his or her genetic profile, and then goes and buys extra insurance because of that knowledge. Well, who in the end pays the bill? Well, you might say, well, the insurance companies, and they are rich anyway. But that's not really true. Where do the insurance companies get their money from? They get it from premiums that everybody else pays. So privacy is not so simple. One is instinctively inclined to support that idea, privacy of medical information, but at the same time it's not so simple because it can lead to extremely inequitable effects or consequences in the insurance system of which we are all a part. This is a very complicated subject, and I don't have to go into it more. I can't go into it more right now, but perhaps we can get to it in the discussion. Suffice it to say, it seems to me that it is important not to become absorbed with exaggerated fears that the Human Genome Project will foster a drive for the production of super babies, or indeed, on the other side, for the callous elimination of the unfit. It is essential, it seems to me, to focus on the genuine social, ethical, and policy issues, some of them already evident, that the Human Genome Project raises, and to respond to them by creating codes of law and/or regulation for the use of human genetic information by geneticists, the media, insurers, employers, and the government itself. Thank you.
You've now heard the formal remarks of all of our presenters. They've given us a lot to think about, to question, maybe even to disagree with. We'll be taking a 10-minute break, and we urge you to take advantage of the 800 number on the screen to share your questions and comments with us. We will be back in 10 minutes.